Hey guys. So today I'd like to talk very briefly about the process of protein synthesis, which is basically how our DNA, our genes, code for our traits. Right? We all know that DNA is the instructions that make us individuals, right? So like I have blue eyes because my DNA is blue eye DNA. Right? But this is the process by which that actually occurs. Right? So protein synthesis is how genes turn into traits. And so DNA is effectively the instructions for the proteins that actually determine our traits. So DNA is just instructions. Right? It's the actual proteins that have the qualities that our traits portray. Right? And so the phenotypes, the expression of our traits, are controlled by the proteins themselves. So regions that code for these proteins are called genes, and our entire collection of genes is called our genome. And our genome is basically just this big book of instructions for all of the different proteins that we need to be us. A big old book full of all those instructions. And so the process of DNA transcription into RNA, and that RNA is translated into a protein, is called protein synthesis. Okay, so we have transcription and we have translation, are the two processes that occur that bring us from the instructions of DNA to the actual protein enacting those instructions and giving us the trait. So we have DNA gets transcribed into RNA, gets translated into proteins. Now, transcription, the process that takes us from DNA to RNA, it's fairly simple. Right? We have an enzyme called RNA polymerase. The RNA polymerase splits our DNA strand apart at the gene for whatever protein we're going to be making. Okay? So our DNA is double-stranded. RNA polymerase opens it up. Okay? It then slides in and it reads the DNA. Okay? And so the RNA polymerase will hook on to the nitrogenous base of whatever nucleotide it's at. It will read it and it will find a complementary nitrogenous base nucleotide for the RNA. Okay? So if the DNA has an A, the RNA polymerase will find a U. Okay? If the DNA has a G, the RNA polymerase will find an RNA-C. And it'll go all the way down and create this complementary RNA strand. So RNA is very similar to DNA, but it's single-stranded. Once the RNA strand is complete, the DNA closes back up, perfectly fine, and the RNA polymerase moves on to find another gene that needs to be transcribed. This RNA is now floating around inside the nucleus, and it's trying to leave to go on to the next step. So here is just an example of the complementary base pairing. Again, A in the DNA pairs to U in the RNA, T pairs to A, C pairs to G, G pairs to C, A pairs to U, and T pairs to A. Okay? So this gold would be our new RNA strand that's about to float away. The white is our original DNA. Our RNA then exits the nucleus, right? Floats away, says, bye, I'm out. Right? And it's going to find its next friend, the ribosome. Yeah? And these ribosomes live on the endoplasmic reticulum that is right next door to the nucleus. It doesn't have to travel far. It's like, boom, here I am. Let us make some proteins today. Okay? And so these ribosomes stud the rough endoplasmic reticulum, the rough ER, okay? And the RNA goes and finds one of these ribosomes. Okay? That implies intent. Actually, the RNA is just floating around willy-nilly, and hopefully it ends up hitting the ribosome. It will, right? That's how it works. But it's not like the RNA is actually hunting out the ribosomes. Okay? So the ribosome's looking kind of like a hamburger bun, has two parts, has a large subunit and a small subunit, top and a bottom. Okay? And the RNA slides in between the two of them. So the ribosomes are what are responsible for actually doing the translation process of these things. Right? If we talk about just ribosomes, we say their job is building proteins. They build the proteins using the instructions found in this RNA. So if DNA is a big book full of all of the instructions, right? RNA is like we took that big book, we opened to a very specific page, and we made a copy of that page. And we take our copy with us, and we go to the buildy place. Right, so now I have my copy for just my one specific instruction. Right? I bring that instruction to the ribosome, and the ribosome is going to build me this protein. Okay? So the ribosome is going to be reading the instructions on the mRNA, which is the RNA that we've sent out into the world. Okay? And it's going to be reading those instructions in groups of 3. So we call these groups of 3 nucleotides a codon. Every codon matches with a specific amino acid. Okay? And amino acids are the building blocks of proteins. So if we look inside the ribosome, we would actually see that there are 3 chambers. Okay, so we're looking at this model. I got my 3 inside chambers of my ribosome. I got this cute little guy floating around over here. Okay, this is a tRNA, okay, also a type of RNA, but this one stands for transfer RNA. It's transferring amino acids. Okay. And so this transfer RNA has an amino acid stuck to it. Okay. Specific transfer RNAs grab specific amino acids. Now in my model over here, I've already had 5 codons processed by this ribosome. Okay, so I got codon 1, 2, 3, 4, 5, and I have 5 amino acids coming out of the ribosome in this chain right here. Okay. Each of these amino acids was coded for by one of these codons. Okay. So the 3 chambers are engaging with codons. Okay. In the first chamber, the codon of the mRNA connects to the anticodon of a specific tRNA. Okay. Once that happens, the RNA moves forward. Okay, so that moves into the second chamber. The second chamber rips the amino acid off of the tRNA and adds it to the growing chain of amino acids coming out the ribosome. And the third chamber ejects the naked tRNA. Okay, boom, bye tRNA. He has to go find his amino acid again. So each of the codons of the mRNA is matched with a specific anticodon of the tRNA, and that allows the ribosome to find the correct amino acid for the instructions. Okay? So if this is my codon right here, AUG, my anticodon is the complementary base. Right? So A goes with U goes with A, G goes with C. So my anticodon would be UAC. And this UAC tRNA is always going to hold the same amino acid. In this case, it's methionine, right? So methionine is always associated with UAC as an anticodon, and therefore always associated with AUG as a codon. So anytime I have AUG on my mRNA, I'll have a methionine in that spot for the amino acids. Yeah. And so the ribosome takes all of the instructions from the mRNA, matches it with the tRNAs in the appropriate spots, and builds this long chain of amino acids. This long chain of amino acids that's coming out the ribosome butt, right, is going to eventually fold up into a very specific ball. That specific ball has a very specific shape, and that very specific shape gives its very specific function. Right? And so if this ball is, the melanin in my skin, right, that shape is going to be brown to give me more brown skin, right? And that's how that works. So the protein possesses the actual characteristics of the trait, whatever that trait might be. And that's it. That's how we go from having DNA as instructions to actually having physical traits that have phenotypic representations. Cool. Bye.